immunodeficiency get classified into primary immunodeficiency, and the secondary immunodeficiency, primary immunodeficiency and secondary immunodeficiency. Okay, so secondary immunodeficiency. Okay, so primary immunodeficiency. In case of primary immunodeficiency, the primary immunodeficiency is the disorders which are caused by the defective genes. That immunodeficiency disorders which are caused by defective genes. That means we are talking about genes, means that will be definitely inherited issues here. We have an example here, that severe combined immunodeficiency disorder, severe combined immunodeficiency disorder, which is by the defect of a gene called ADA gene. Okay, so we'll discuss in applied biology more. The defective gene, severe combined immunodeficiency syndrome, that because of a defect in the gene called ADA gene. Okay, so what do you say? Primary immunodeficiency disorders are caused by the defective genes, like severe combined immunodeficiency syndrome, which is caused by the defect of a gene called ADA gene, adenosine deaminase gene, we use it to say. And the secondary immunodeficiency disorder. The secondary immunodeficiency disorders are caused by various factors like infections, aging, the different viruses even can cause that. We have a best example for that, HIV, AIDS. Okay, so deficiency, definitely we know that AIDS is the immunodeficiency syndrome, then, but it is secondary immunodeficiency syndrome. That means it is acquired one which will come after the birth. That is very important. It won't come through genes. It is not primary one. It will come after the birth because of viruses. Okay. So not through the genes. At least you need to remember like this. This is not through genes. This is through viruses. This is not heritage disorder. This is not at all heritage disorder. Okay. So that you need to remember here. Now, what is AIDS? That well known, acquired immunodeficiency syndrome. AIDS means acquired immunodeficiency syndrome, which is caused by that HIV virus. That AIDS caused by HIV virus, human immunodeficiency virus. Human immunodeficiency virus, we use it to say here. So that direct effect of this particular virus is by itself immune system, that their target is itself immune system. Let's take a look on this. That acquired immunodeficiency syndrome, it is transmissible, mostly sexually contact disorders. It is the most, it will come by sexually transmitted disorder it is. And it's a lethal, what is lethal? Very harmful, we call lethal disease, caused by human immunodeficiency virus, HIV. We use it to say it as HIV. Okay. First reported by 1981 by Center for Disease and Control. The 1981 by Center for Disease and Control of USA, and in the last 30 years, it has spread all over the world, killing more than 25 millions of people. More than 25 millions of people are killed here because of HIV. 1981, Center for Disease Control people identified this, and in the mean period of time, it is making its own mark. So let's continue. The HIV, acquired immunodeficiency syndrome, caused by human immunodeficiency virus. That mode of infection of this particular disease, how it spread. It generally occurs by sexual contact. The disease will come by sexual contact with an infected person, by transmission of the virus contaminated blood, by sharing infected needles, and from an infected mother to her child through placenta. You just please notice this, that infected mother to her child through placenta. Earlier I said this HIV is a disorder which is not, we are not going to say this is herited disorder. Okay, non-herited. So by showing this, the students will ask here, sir, infected mother to her child. So that can't you say it as heritage disorder? No, even you can't say it as heritage disorder. Because you just notice what the word is given through placenta, not the genes. Through placenta. That means through the fluid transfer that is. So mother is infected through the placenta, it is going to infect the child, not through the genes. Okay. So that you need to remember. For example, if you take an example here, father is HIV patient, mother is HIV patient. Both are HIV patient. Then in vitro condition, if we collect the sperm from the father, ovum from the mother, and in the laboratory, if you do the fertilization, that means a zygote is formed in the laboratory in vitro fertilization. We are well known for learn in reproductive health. And if we introduce this particular zygote in embryonic stage in surrogate mother, so do that particular child will get AIDS? No, not at all, because that won't come through the genes. That you need to understand. Okay. So from the mother to the daughter, it will come not by genes through the placenta. So that's why it's definitely it is secondary immunodeficiency. It cannot be a primary immunodeficiency. Okay. It is important to note that the HIV/AIDS is not spread by mere touch or physical contact. So let's talk about the structure of HIV. Yes, you may know a particular virus don't have a specific structure. Whatever the structure for corona or HIV, whatever we are showing, that's going to be a hypothetical diagram only in general. Okay, so the HIV is a retrovirus. Remember, HIV is a retrovirus along with hepatitis B. Both are retroviruses, which has an envelope closing two single-stranded RNA. See this, two single-stranded RNA. Two single-stranded RNA are present with a reverse transcriptase enzyme. Single-stranded RNA as a genetic material they have, and two molecules of reverse transcriptase enzyme. You see here that above there only you will find a reverse transcriptase enzyme. So this is reverse transcriptase enzyme. To try to identify this diagram, I am exposing. Okay. Two single-stranded DNA with reverse transcriptase enzyme. The two molecules of the enzyme reverse transcriptase. The single-stranded RNA. So the structure we started from inside. The single-stranded RNA is the single-stranded RNA surrounded by a protein coat. Just follow the theory with the diagram. Single-stranded RNA is surrounded by a protein coat. So I am showing here. This is the protein coat. This is the protein coat. Single-stranded RNA is surrounded by a protein coat, followed by a layer of proteins, which again surrounded by outer lipid layer. So followed by a layer of protein. So I'm showing here again, followed by a layer of protein. Okay. Then a layer of protein, then by outer lipid layer that you'll find outer lipid layer. So this is a lipid layer. Lipid layer is there. Okay, so that contain a number of glycoproteins, GP120, GP60, GP80, GP140, likewise, the glycoproteins, molecular, GP41, GP40, likewise, GP means glycoprotein. Okay, so that is the structure. These proteins bind to the host cell surface receptor. These GP proteins are going to be bind with the host cell surface receptor. Let us see the diagram in Zoom that will understand clearly. Text diagram itself it is. Okay, so structure wise, here it is. In maximum zoom, this is enough, I think so. Yeah. Okay, now then on full screen you can see the diagram. On full screen, is it more possible? No. Oh my God, we lost it. Yeah, we got it back now. Now it is virus on full screen. Okay, so just follow the norm. Just follow the norms. the first of all, we have to start from the inside onwards. Inside that you can see the single-stranded RNA with reverse transcriptase enzyme. This one, single-stranded RNA with reverse transcriptase enzyme. So then after what we said, that there is a protein code. There is a protein code is present. Single-stranded RNA with reverse transcriptase enzyme with a protein code. Okay. Next, that some layers of proteins again. Some layers of proteins again. Okay, so that is given as layer of protein. Next, after that, what we can see that a lipid layer. That after that, a lipid layer is seen over. Violet color is shown here. Let me try to take the same. No, not possible. Yeah, this is the lipid layer. I'm closing that area. Lipid layer. Okay. So above on the lipid layer, you can see the glycoproteins. These are the proteins. Glycoprotein, GP20, GP40, GP41, GP60, GP120. Likewise, the various kinds of glycoproteins are present here. That's the story of this particular. Just while reading itself, you make your own numbers that make you to be helpful for without reading the theory, you can understand. So we have to start from here. This is the number one, single-stranded RNA, okay, surrounded by a protein coat, that is number two. Next, number three, we have to make, that means some more layer, yeah, no, is there here, layer of proteins, number three. Next, a lipid layer, number four. Number five is the glycoproteins. Number five is the glycoprotein. If you make your own numbers like this, it will be helpful. Easy, very easy. You can make it the structure. You can draw the diagram. Diagram that not necessary here. This kind of three-dimensional diagram, just in your own. That we have to make circles. Likewise, you remember that you can draw the diagram. Okay. So that's it. And the structure of HIV virus is so easy for you if you put the numbers like this. Okay, numbers like this, it will be very easy to. If it all possible, you take the screenshot and make it in your text afterwards with the same numbers. Okay. Next, let's continue. Let's continue. Then let's go back. So the structure of HIV we are seeing now. Structure of HIV, the mechanism, how this particular parasite is going to affect, that what are the particular parasitic issues when it enters into a host cell body, that means itself human being. So what makes that, what kind of mechanism we have. So after getting into the body of a person, HIV enters the T helper cell, macrophages, or dendritic cell. Did you remember? These are very important. That means knowledge of immunology will be definitely helpful for us to understand these particular issues. So what we say that first of all, first they are going to affect the T helper cells. Okay, so T helper cell and T helper cells they are going to affect. What is the function of T helper cell? We are seeing T helper cells are for both humoral immunity and cell mediated immunity. They are going to present that the microbes to the respected police, that both B lymphocytes and T cytotoxic cells, isn't it? So that means here straight away it is affecting on them. That did you remember the macrophages and dendritic cells? These two cells, macrophages and dendritic cells, we call them as APCs, antigen presenting cells. So that means the greater effect is here itself, the greater effect. So first, after getting into the body of person, HIV enters into the T helper cell, macrophages, or dendritic cells. Okay. So what happens immediately? What happens immediately that you can understand, the person lose the capacity, that person, that host, that means patient, we can say, he lost the capacity of recognition. So he lose the capacity of recognition of the various kinds of microbes, capacity of recognizing, because these are the pioneers of the helper cell, macrophages, dendritic cells are the pioneers which they are carrying that particular microbe. They are representing that, identifying the microbes and presenting to the respected immune competent cells of our body. Okay, so if they lose the capacity, just like that, informers are lost. So informers are lost, that means it's very difficult to the police to identify the thieves here. Informer means a person who informs he is the thief, he is the terrorist, likewise. So that informers will lose. Okay. So immediate HIV virus when it attacks. Okay. So host patient lose the capacity of recognizing microbes. So that is the immediate effect you need to understand. Okay. So let's continue. In these cells, the single-stranded RNA, when it enters into the cell, the single-stranded RNA of HIV synthesizes a DNA strand complementary to the viral RNA. That means it enters in the cell, the single that HIV virus. Then after that, what it has doing single-stranded RNA now becoming a DNA strand complementary to the viral RNA. That means single-stranded with the help of the reverse transcriptase enzyme which is present in them, so that it will turn into the DNA. It will turn in the host cell. This is the host cell. That means it may be helper cell or macrophage or the dendritic cell. So that is turned into that particular DNA strand. Okay, using the enzyme reverse transcriptase. So reverse transcriptase also catalyzes the formation of second DNA strand complementary to the first strand, forming the double-stranded viral DNA. So finally, what is formed here? Double-stranded viral DNA is getting formed. So simply you need to understand what is the function of the reverse transcriptase enzyme which is present on the single-stranded RNA, making that single-stranded RNA to the double-stranded DNA. Okay, so now what happened? Viral DNA, double-stranded DNA is formed. Okay, so this viral DNA gets incorporated into the host cell DNA by viral enzyme integrase, and now it is in the form of provirus. This particular DNA will be incorporated into the human DNA. Okay, so let me take that. Let me erase this. We'll show this where it will go. I'll draw a little bigger host cell that you can understand. So imagine this is the host cell. Okay, this is the host cell. Now a single stranded RNA now became as a double stranded. So I'm drawing the nucleus. Okay, so nucleus, we have our own DNA, isn't it? We have our own nucleatic material, 23 pairs. Now what I am saying, that these particular single stranded RNA, after turning into the double stranded DNA, it will go and settle down that into the host cell nucleus. Okay, so the viral DNA gets incorporated into the host cell DNA by a viral enzyme with the help of an enzyme called integrase. Integrase enzyme makes it that it makes it incorporating into the host cell DNA. Okay, so red one is our DNA, host cell DNA. This is the DNA of HIV virus, the DNA of HIV virus. Okay, then this is the story. Okay, so what happens afterwards? So who makes them to go inside? Integrase. Now onwards, this particular HIV viral DNA is considered as provirus. It is going to be considered as provirus. Okay, so this is called provirus. Okay, so that green one, which I'll write here, that you can understand. Provirus. It is a bigger factory then. The transcription of the DNA results in the production of RNA, which can act as a genome for the new viruses, or it can be translated into viral proteins. Okay, so that means the virus, various components of the viral particles are assembled and the HIV are produced. Infected human cells continue to produce the viral factories, and in this way they act like HIV generating factories. Now, this DNA is going to produce a lot of single stranded RNA. Okay, so that's why we are going to say it as the factory, HIV generating factory. And also the genome it has, that particular nucleic material it has, it also produces the various kinds of protein. So only they have formula. That means you may lend over in genetic code. Nuclear basis sequence makes over protein synthesis. So they have only sequence of code. All the ingredients will be taken from the host cell DNA itself. All the ingredients are taken from the host cell nucleus itself. So that's a magical thing here. A single stranded viral RNA entered, and with the help of reverse transcriptase, it became that DNA, and with the help of integrase enzyme, it enters incorporated into the host cell nucleus, and there onwards it started the producing the various kinds of proteins and the many number of the viral RNA. So that is going to be that HIV generating factories, we can say. New viruses bud off from the host cell. Whatever the new viruses are produced here, they bud off from the host cell. Okay. So this lead to the progressive decrease in the number of T cells in the body of infected person, leading to the infected person, leading to the immunodeficiency. Slowly, that the new RNAs which are produced from this particular factory are going to be the enters into the new helper cell, and there also they do the same with reverse transcriptase. It become DNA and become with the help of integrase enzyme that going to become a part of our DNA. So then again, factories are increasing. So that brings the various effects here. So first of all, let me see that they travel. Then after we'll see what it affects, how it affects. New viruses bud off from the host cell. This leads to a progressive increase in the number of T helper cells in the body of an infected person, leading to the immunodeficiency in him. Even though HIV attacks any cells with CD4 markers, that for reasons not known, how why only that T helper cells are going to be destroyed, that not the macrophages, that we don't know. Okay, here CD4 means T helper cells. That means that they are attacking T helper cell, macrophages, dendritic cell, but we don't know the reason why they are going to destroy only T helper cell, not the macrophages. They are attacking, entering inside, but they are destroying only T helper cell. Okay, with that, those who are having CD4 markers, we call T helper cell. If you remember, we call them as T4 cells or CD4 cells. Okay, the GP120 molecules present on the surface of HIV, the GP120 molecule present on the surface of HIV attached to the CD4 receptors of the human cells. We have seen earlier now in the diagram, GP120 glycoprotein. So these glycoprotein GP120 molecules which are present in the that HIV virus are going to connect it to the T helper cell CD4 markers. We said T helper cells are having cluster differentiating 4 markers are there. This GP120 is going to attach with them. Okay. So that's a story that mostly that maybe that is the reason that mostly T helper cells are going to be attacked over. GP120 of the HIV virus fits on the CD4 cluster differentiating molecule. Okay, that to be remembered. Attack on the certain types of cells or tissues only by viruses such as HIV is referred to as tissue tropism. So that means particularly targeting a particular cell or tissue of our body by viruses, we call it as tissue tropism. Okay, what is tissue tropism? For that specificity, a virus specifically acting on one particular cell, like HIV virus specifically acting on the T helper cell and destroying. We call that feature as tissue tropism. We say that feature as tissue tropism. Okay. Then next, let's continue. This is the effect actually that what we said. The mainly they are going to be affected on helper cell, macrophages, dendritic cell, and we said they are going to destroy the helper cell. So what happens? What happens? That we need to see in the form of symptoms. There is always a time lag between the first infection and appearance of the symptom. There is always a time lag between the first infection and appearance of the symptom. So this period may be few months to many years. It may be usually 5 to 10 years even. It may that particular parasite may take time to develop into fully blown AIDS after the infection. Okay, so during this period, the person suffer from bouts of fever, diarrhea, loss of weight. These are the general symptoms. 10% loss of weight, we can say that he may have, you can suspect in that person. Okay, loss of weight, some cases will happen, but 10% sudden loss of weight may be indication of this particular. Why? Why diarrhea? Why bouts of fever? Why that fever again and again comes. So that you see that major mechanism, helper cell macrophages, dendritic cells became non-functional now. That means welcome to all the other microbes and that which can cause the disease, which can affect the human being. So that means our police informer system is destroyed. That's why the police may not recognize. That's why various kinds of opportunistic infections, we use it to say them as various kinds of infections may come. Okay, so that is due to the decrease of number of T helper cells. The person starts suffering from infections due to bacteria. Anything, some examples are given, like especially mycobacterium, viruses, fungi, and other parasites such as toxoplasma. Toxoplasma is protozoan parasite. Toxoplasma gondii is a protozoan parasite. So the patient becomes so immunodeficient that may are unable to protect themselves against the minor infections, which normal healthy people can easily overcome. But these people with minor infections, even there's going to be bigger to them. That's why, because for all that we have answer. That's why, because the T helper cell, macrophages, and dendritic cells are infected by the virus. That's why they're not working properly. That's why any kind of micro protozoan or bacteria or virus or fungi that may cause the infection. Okay. The time between the first exposure to the HIV and the production of antibodies by the immune system in response to the HIV infection is called window period. Define window period, that means that time between the first exposure to the HIV and the production of antibodies by the immune system in response to the HIV infection is called window period. We use it to say it as window period, during which the time the presence of virus cannot be detected. In this particular period, we can't find out that we cannot detect that particular virus. Okay, so that's it about the symptoms and window period. Diagnosis: how do we recognize? How do we recognize that we have the best way? That ELISA is a primary the technique we can say here. That the exact test will be what we can say. Wait a minute. How to erase? Yeah. Then diagnosis. A widely used diagnostic test for detecting HIV infection is the ELISA, enzyme-linked immunosorbent assay. We use it to say that we are going to discuss in detail ELISA. ELISA test, how it can be done. The various types of ELISA, direct ELISA, indirect ELISA, in that sandwich ELISA. We are going to discuss all the issues in the coming next class. Then ELISA, enzyme-linked immunosorbent assay. But in general, we can say it's just screening test. The sometimes positive, even maybe false positive or maybe false negative can come here in this particular case. Okay, but if you want exact confirmation, Western blot technique. Western blot is used for more reliable the confirmation test for HIV infection. So screening test you have to remember as ELISA, enzyme-linked immunosorbent assay, and Western blot is used as a more reliable confirmation test for HIV infection. So that's it. These two tests we have to remember. Our HIV, which can be detected within 15 days to 4 months after the exposure to the virus, because ELISA test based upon the recognition of antibodies mostly, that we have antigen type also, but in case of HIV, we are doing the test for detection of the antibodies in general. This is simply antibody test. I mean, nowadays people know what is antibody test. Okay, ELISA is the antibody test that we'll discuss when we go to the next class. ELISA, how it can be performed with the videos will show. Okay, so the widely okay, then treatment, how, what kind of treatment can be done for this treatment of HIV infection and prophylaxis. Okay, so treatment aids antiretroviral drugs, zidovudin, azidothymine, didaxinosin, didaxicitidine. Nowadays, the medicines which are going to be used, we say simply as antiretroviral drugs can only prolong the life of the patient, but we can't say here, we cannot prevent the death. Okay, hence it is better to follow prophylactic method. That means not to come at least. Prophylactic method to avoid HIV infection. Simply we are saying there is no proper treatment for the complete cure, but at least we can say, use antiretroviral drugs and some drugs like zidovudin, azithromycin, redaxinosin, dedaxycytidine. So these kind of drugs will prolong the life of the patient. Okay. Prophylaxis. That prevention we say simply. Prophylaxis nothing but prevention. Okay. So how do we prevent this particular parasite? How do we prevent? Prophylaxis or prevention, nothing but prevention. You just understand. Prophylaxis is nothing but prevention. Okay. So how do we prevent? Safe sex, proper sterilization of the hospital syringes and needles, and screening the blood for HIV before transmission are some of the most important prophylactic methods here. In our country, the National AIDS Control Organization, NACO, NACO, National AIDS Control Organization, and other NGOs, national non-governmental organizations, are doing a lot to educate the people about AIDS. Okay, so that's the story of this particular area. That not so difficult here. We are not learning the complete technical information. This simply what we say. We need to learn the concept which is gives the minimum knowledge, minimum information about the HIV virus here. So the major things, major points to be remembered here in prophylaxis, that about the people of NACO and NGOs, treatment, antiretroviral viral drugs can prolong. Okay, the diagnosis wise, ELISA and Western blot techniques. ELISA is a screening, and for perfect confirmation, that Western blot technique. Okay, in the case of the symptoms, that person to person, some window period may be, incubation period may be different here after the first infection and appearance of symptoms. We call incubation period. Then the primary infections like bouts of fever, diarrhea, and loss of weight, it may come, but in future time it may go affect the lymphoid organs also, which leads to the complete immunodeficiency. The person is going to die not because of virus, because of what here? Because of opportunistic infections. That means virus just destroyed the police station. That's all. Okay, so what happens? The leftover thing is done by various microbes like bacteria, viruses, fungi, and the protozoan parasites like toxoplasma. We say all them as opportunistic infections that do the rest of the work here. Person immune system becomes so weak that any infection it will come to the person. So in this area mechanism, we learn the structure of HIV virus and their nature. Okay, nature wise, helper cell, dendritic cell, macrophages they like actually, and GP120 of the HIV virus glycoprotein GP120 is often fit to the CD4 markers of the T helper cell. That kind of specificity we say that tissue tropism. That is we learned here, and the two enzymes, the reverse transcriptase and integrase enzyme. One makes a single stranded to the DNA and later makes that incorporation of that double-stranded DNA, which is the viral double-stranded DNA, can be enter into the nucleus of host cell. Okay, the structure-wise, single-stranded RNA with reverse transcriptase enzyme, and which is surrounded by that particular protein coat and layers of protein, then the lipid layer and the glycoprotein lollipops. You'll find surround here GP20, GP40, GP41, GP60, like with the various glycoproteins can be expected over. So that's it about the story of HIV, AIDS. Okay, so AIDS or HIV virus. So that's the story of HIV virus. And then we continue with the various other issues which are left over in immunology.